plutonium is basically breaking into uranium and other elements. But the other thing that is quite interesting in this description is that the effects are on the nucleus of the atom. And actually, even cells have their nuclei. So this is again motor neuron. This part is what is called the nucleus. And nucleus is the site in the cell that actually contains all the genetic information. The 3 billion of letters of our DNA are packed into this small structure, the 6 feet of the double helix strand. In these 3 billion letters, they encode 20,000 different genes. And what are genes are stretches of DNA that basically define the sequence of proteins, the amino acid sequences that I described before. And so what differentiates one cell type, motor neuron, from your blood cells? Actually, it's not that each cell would have different set of genes, but in each cell, different set of genes is turned on and off. So basically, each cell selects only a subset of these genes to turn them on, and that defines the function and identity of individual cells. So if you look at a piece of a chromosome or DNA strand, so this would be maybe something like 1 million of these DNA letters. You see that within this region of the DNA there are several different genes, and as I said, in each cell a different group of these genes will be turned on. Now, how do cells actually control the genes, which gene they will turn on or off? That's, to me, like the most beautiful problem in biology, because actually cells use products of genes to control their own genome. And these genes are called transcription factors. They are proteins that actually can recognize sequences in the DNA and can actually find these sequences among the 3 billion nucleotides or DNA base pairs in the genome and sit at very specific locations. And so you can also notice that most of the DNA does not contain genes. It's this empty space, and that's exactly where these transcription factors bind. We have around 1,639 transcription factors as the latest count. And basically, which of these factors are turned on in a cell will define which regions of the DNA they will occupy, and then also which genes they will regulate. This sounds sort of very simple and mechanistic, and suggests that we should be able to predict how transcription factors control DNA, but in reality, actually, it's not the case. So you can think of these transcription factors as these little fields of response, because they can, when you put them in the nucleus, they will change the organization of the nucleus, the structure of the nucleus, and they will change the identity of cells. But the effect of these transcription factors is still really unpredictable for us. We cannot computationally say that adding this transcription factor will convert skin cell into a motor neuron, for example. And so a lot of these studies relies on empirical investigation, where we add one transcription factor to a cell and ask how cell responds, or take the factor away and ask how cells respond. Similar like the alchemists when they were mixing different molecules and heating them and seeing how the metals respond. So actually, we were quite interested in understanding what transcription factors would turn on the genes that define motor neuron identity. And that's work that was done from a postdoc in my lab, Mr. Armazoni, who's now at NYU. And he basically, as an alchemist, started testing different transcription factors that he added to embryonic stem cells, these pluripotent cells, and asked him whether he can find a combination of factors that would reprogram or transmutate these embryonic stem cells into motor neurons without relying on the normal developmental pathway that we relied on before. And he actually focused his attention on two factors. So this is again a cross-section of the developing spinal cord. This is where motor neurons are born. And there are two factors that are very highly present or turned on, specifically in the location where motor neurons are being born. And these transcription factors are called ILAD1 and LHX3. And remarkably, when Esteban built and engineered an embryonic stem cell line in a way that he can turn on these two transcription factors in the cells at his will, it turns out that when you activate these two transcription factors, within 48 hours, so very rapidly, these cells basically convert from rapidly dividing small cells into motor neurons. And it happens very efficiently and also very rapidly, which was quite remarkable because that shows us that we actually identified the key factors that can turn on the right set of genes in order for these cells to become motor neurons.
Mm-hmm.
So this was great, but yeah.
I'm so sorry.
No problem.
You need both transcription factors?
Yes, you need both factors.
One or the other doesn't do it?
No, one
They
or
need to be added sequentially?
So we have not tried to add them sequentially, but we know now. So we also mapped how they bind to the DNA, and we know that they actually bind together to the same sites in the DNA. So we think that they form actually what we call heterodimer. They bind to each other, and then they sort of identify the right locations in the genome to activate the genes. So this was really exciting because it showed us that eventually we might be able to find combinations of transcription factors to produce almost any cell type in our body that could be of clinical relevance. But eventually we actually turned out to another question, and that question was related to the fact that, or to the question of why motor neurons are susceptible to disease only when they reach certain age. And so that brings me to the second part of the alchemy, which is the elixir of use. So I told you that transmutation happens relatively, it's something that happens in biology, in nature, and it's relatively easy to justify. But elixir of use was always something that people joked about, that alchemy never actually showed that something like that could exist. But we think that with these transcription factors, we might be might be eventually able to actually change not only the type of cells that we produce, but also age of these cells. And so this is work that started in my lab by talented postdoc Tulsi Patel, who just last summer moved to her own to start her own lab at Rutgers University. And what she asked is how motor neurons change from the time they are born early in the embryo all the way in aged organisms. And she asked how these motor neurons very quickly do they turn on the right set of genes, and as soon as they turn on the right set of genes, they maintain the same set of genes through the lifetime, or do these cells change as the organism matures? And so she isolated motor neurons from all these different stages from mice, and then asked what genes are turned on or off in these cells. And so now I'm representing her data in sort of this more abstract way, where you basically can ask which of the 20,000 genes are expressed, and then just show distance, how different the expression is from one state to another. And so this would be how the cells in the embryonic day 10 mouse looks. This is 3 days later, embryonic day 13. This is now postnatal day 4 motor neurons, postnatal day 13 motor neurons, and then postnatal day 21, 56, and 2 years. And so what you can appreciate here is that there is a remarkably dynamic change in gene expression all the way from embryonic day to postnatal day 21. And at this point, at postnatal day 21, the motor neurons become very stable, and they do not really change during the rest of the adult life. And so that suggested to us that there has to be some kind of program, gene expression program, controlled by some juvenile transcription factors that activates this youthful program in motor neurons. And then there has to be some adult program which is activated by postnatal day 21 in these animals,